The sliding filament theory. The sarcomere is a structure of myofibrils in muscle.
Myosin as well as actin are the contractile filaments of muscle.
The muscles contract when thin and thick filaments slide past one another.
Myosin actually pulls the actin. Cross bridges of the myosin filaments attach to the actin.
This is the sliding filament mechanism. A muscle contraction begins when ATP is broken down into ADP and phosphate. This causes the myosin hand to extend and form a cross bridge.
The myosin pulls actin towards the M line, shortening the sarcomere.
ADP and phosphate are released when this happens. The myosin stays attached to the actin until a new ATP binds to the myosin. The myosin then goes through another cycle of binding or remains relaxed.
Muscle contractions are controlled by the actions of calcium. Calcium is associated with proteins called troponin, shown in green, and tropomyosin, shown in pink.
When the muscle is relaxed, tropomyosin blocks the cross bridges. Later, calcium ions bind to the troponin, which displaces tropomyosin to form a cross bridge.